Hello, everyone, and welcome to today's webinar titled Extended Release Injectables. This is the third of a series of webinars hosted by Ashland in order to maintain contact with customers during this challenging time. At Ashland, scientists are always solving for formulators, manufacturers, and patients. Its pharmaceutical team prides itself on advancing drug delivery with sophisticated science-based solutions. Ashland's global centers of excellence contain the latest equipment, and research scientists and formulators are ready to work with you from conception to commercialization. Ashland solvers work to meet formulators' needs by providing an unparalleled range of excipients and technologies, as well as long-standing polymer expertise and technical support for oral solids, liquids, topical, and parenteral dosage forms. Ashland solutions include increasing the solubility of poorly soluble APIs, controlling drug release of modified release products, reducing tablet size while maintaining API load and tablet integrity, reducing disintegration time of tablets, and enhancing processability. To learn more, please visit www.ashland.com. Today's presenter is Dr. Sean McMahon. Dr. McMahon is the Business and Technical Manager for the bioresorbable polymer portfolio at Ashland, supporting customers with key excipients and technical solutions for the development of extended release parenteral drug delivery formulations, medical devices, tissue engineering scaffolds, and cosmetics. He joined Ashland as part of the Vornia acquisition in 2018, where he previously held responsibility for the company's major business function in his role as CEO. During his time at Vornia, Dr. McMahon led the development of the company's major intellectual property platform for production of bioresorbable polymers. He has extensive knowledge of bioresorbable polymer design, chemistry, and processing technologies.
Okay, I think with that I can take over. So I think we had some background introduction there from some other guests. So for the presentation today, I'm going to take us through extended release injectables. So I hope many of the listeners will know what those types of technologies are, but if you don't, take advantage of today, try and understand what they are. And by the end of the presentation, everybody should be in position to understand what injectable technologies exist in terms of extended release formulations, how you control those extended release formulations. So how do you design, for example, for 1 week of extended release versus 1 month versus perhaps 6 months of extended release formulation. In terms of the strategies that exist on the market, I'll talk about the purpose of these technologies, so why we use them, what kind of challenges we're trying to overcome, and then more practically, consider the raw materials that you have in your hand. For PLGA, for example, the most commonly used bioresorbable polymer for extended release systems, it's a thermoplastic, so you can melt it and re-solidify it. It can be provided in powder form, granule form, film form, filament form, all types of form. With your API, you could combine it into different formulation formats, but what do these actually look like in terms of products on the market today? And there are 3 that we'll talk about. These are the 3 that all can be characterized within. And from there, with an understanding of the formulation formats, so what the actual commercial products can look like, we'll take an actual look at the commercial products in the market, some formulation examples, and from there, everyone should understand how chemistry is selected. So for PLGA, for example, you should recognize what type of chemistry features would be apparent in 1 week systems versus 6 month systems. And finally then, based on the chemistry and knowledge and understanding of polymer, we'll take that through a processing example and show you how you could formulate effectively and develop a process and optimize it towards a hot melt extrusion case study. So a little bit on Viatel bioresorbable polymers before we start. Ashland supply a family of bioresorbable polymers for drug delivery, medical device applications, tissue engineering, and cosmetics industry. Today we're focused on drug delivery, so I'll mainly be talking about the amorphous copolymers of lactide and glycolide, so PLGA and PDLLA are the more commonly used ones. The reason for these polymers being used so commonly in the industry comes broadly from the compliance that's demonstrated, the safety associated with the polymer chemistry and how naturally it works with the body, but more practically for the solutions you're engineering, these are amorphous copolymers. They do not have crystalline regions, so they're very easy to program towards predictable degradation. So for example, as chemists, we can program the chemistry for PLGA, and with experience, we can understand what type of chemistry should degrade over a week versus 1 month versus 6 months. As a chemistry company, we can provide you with that solution, and as a drug delivery company, you combine that chemistry with your API, deliver it in some sort of format into the patient, and you could tune release over the period of time that's required. The other polymers that I won't go through today are polymers that we have within our portfolio. They do sometimes exhibit use in controlled release drug delivery type applications, but they're more commonly used for medical devices. Their duration of degradation timeframe would extend beyond a year, up to maybe 5 or 6 years in the case of these polymers. And this comes back to the structure, the semi-crystallinity that they exhibit, and the higher load-bearing capacity that they have. In terms of Ashland's Viatel bioresorbable polymers, we offer this chemistry from a production process that allows us to deliver no residual solvents. So many processes exist for producing these types of polymers. One common type of process would use a lot of solvents in technology to create and thereafter purify. When working with Ashland's Viatel polymer, we've removed these solvents in terms of residues from the equation entirely. So if you look at the gas chromatography comparison of Ashland's Viatel polymer versus a competitive polymer in the same industry, you can see from Ashland's case that there are no solvents, no types, and no amounts present relative and respective to parts per million. So down to parts per million, we would exhibit 0 as a solvent residue, whereas for a competitive product in the same market, for one such case, you find residues of ethanol, acetone, isopropanol, and toluene. So solvent residues are acceptable to a degree in pharmaceutical formulations, but most will understand that these solvents can cause inflammatory reactions in the body. They may interact with certain drug compounds, and they can influence polymer properties in terms of plasticizing polymers. So it's a solution that we've developed trying to remove any inconsistency that might come into play from these solvent residues. They're an area that you can't control very well if they're present, so they're not part of the product that you're actually seeking to work with, which means if we remove them, you should be able to relay whatever impact you see in the application back to the chemistry alone, which gives you closer contact and control over your formulation. And this ideally will lead to better consistency in terms of the product performance. In terms of purpose of BRPs, and BRPs stand for bioresorbable polymers in pharma, if we take a look at drug compounds, there are many that exist. Some present toxicity, some will be fast or quickly metabolized or excreted through the oral canal. Beyond that, you exhibit different levels of variability in terms of PK, blood concentration of drug compounds after administration through oral canal. And finally, in certain cases, it doesn't always make sense to take a tablet. So for example, if you think about a localized issue, for example, infection in the eye, is it really the most sensible thing to take a tablet, allow the tablet to go through your system, deliver large quantities of that tablet into your entire system to get concentrations after a period of time to the eye? Or would you benefit, or would there be an opportunity for you to deliver perhaps a lower concentration directly into the eye, getting quicker onset and higher concentration at the eye? So with respect to that, take a look at the graph on the right-hand side. What we have is concentration versus time. So there's a window for the therapeutic effect within which you would like to keep your concentration of drug in the bloodstream or at the site where required. We call this the therapeutic window. When taking tablets, you have to manage patient compliance. So for example, you take a tablet and you wait for 8 hours or a certain period of time before you take another tablet. There are all kinds of variations that can come into play within that time frame in terms of patient size, patient gender, what they're eating and drinking, patient compliance in terms of how consistent they are in terms of taking tablets. And in certain cases, it may not be optimal to use tablet-based formulations to develop these technologies. So for example, what we're trying to do with these systems for bioresorbable polymers is take the same drug compound, encapsulate it within a polymer matrix, deliver that polymer matrix containing drug into the body, allow that polymer to slowly break down, and that's the programming step that allows us to tune this release rate and duration. So over a 6-month period, if you were to compare, the first and most obvious advantage is you could deliver one dose versus many, one single injection versus perhaps over 100 tablets. Beyond that, tunable profiles have been achieved. So in the market today, there are over 50 products that use this type of system for controlled release drug products, excluding coatings, so specifically pharmaceutical injectable type products. Within those, they're tuned out to a maximum of 6 months, and there are many products that do that. Beyond that, some drugs exhibit side effects. So if you're dosing via the oral canal through the digestive system, you may have to deliver more of the drug. And in certain cases, you may get a benefit from locally administering. So for example, if I take the eye case again, deliver less of the drug, higher concentration at the site. If the drug has a side effect, you might reduce that and you get faster onset for the actual effect that you're looking for. And then finally, there are certain case-dependent advantages. So some molecules lend themselves very nicely to PLGA type technology. Some reasons are presented based on stability, others based on the fact that some compounds could never go through the acidic climate in the stomach. So taking that all into consideration, these are the drivers that we look at when we consider, would a technology like ours benefit from BRP or bioresorbable polymer type chemistry? So a very brief history on this type of polymer chemistry and how it's been used in medical applications. It's certainly not new to the market, so over 50 years now of precedent in terms of products demonstrating safety and compliance and improvement year on year, new applications and newer products being developed in the market. A number of important products to think about. Dexon was the first, so about 50 years ago, a bioabsorbable suture. Since then, many different products exist. So in 1986, a very important product, Decapeptyl, the first extended release microparticle product. And you'll notice a trend. So there are firsts at several stages, and then after time, there are firsts applying the same technology in a different indication. So for example, recently, Zilretta taking advantage of a similar technology from 1986, microparticle type technology, incorporating a different API and being applied for intra-articular knee delivery. So technologies today can take advantage of everything that's been developed. You can stand on a giant shoulder in a sense, use something that's already been proven, apply it in perhaps a slightly different manner, and get the impact and effect that you want, as well as being confident that these systems are commercially viable and something that regulatory channels will accept with a well-trodden path. So trying to understand how you change your raw materials into final format, you have to be able to conceptualize what it would look like. And so all of the products that exist today will fit into one of these three categories. So in situ forming systems are the first, a very simple system to start with in terms of how you would form a prototype. You have your API, which is dependent on the indication and the company working. The bioresorbable polymer, so Viatel PLGA is quite a common one to use, and a relevant biocompatible solvent. So for the 5 products that use this type of system on the market, the Atrogel type system, they all use NMP as the solvent, and Ashland supply the GMP grade of NMP that's available on the market under the brand Pharmasolv. So you dissolve the polymer within the solvent, and that changes the solid state of the polymer into a liquid state, and this allows you to pass it through a needle and syringe, which would allow you to get past the skin barrier, for example. You load the API within that matrix, so you may suspend it or dissolve it in the matrix, and then inject it. So for example, if I inject it into water, what happens is the depot precipitates, solvent dissipates, and you have a depot forming. So if you do this subcutaneously against some resistance, you can form the depot under the skin. So we use the solvent to get past the skin barrier. Thereafter, the depot forms and should stay in place for the amount of time that we've programmed the polymer to last for. That depot would consistently release drug over the time frame to achieve that therapeutic window that we talked about earlier. Second type of product, very easy to recognize and visualize, a solid implant. So for example, a rod-shaped implant. API and PLGA typically formed by a melt-based process. So you can melt the polymer and incorporate the API. This is typically done via extrusion. These implants, again, certain pros and cons. So some pros exist in terms of the process relating to the fact that you don't need to use organic solvents, so they don't need to be removed, and you can have a clean type implant. Good stability because you have a good understanding of that implant before you put it into the body, and it's not going to change in that initial stage when you implant it. So the burst effect can be better understood from entering the body position. In terms of challenges, these polymers are unstable, so that's a fact, and that's what you're taking advantage of. You're taking advantage of the programmed instability that you wish to use when you're formulating and delivering a compound. But with that in mind, if you're processing these at high temperature, you can degrade them considerably. So you have to be very conscious of that and mindful of that when you program and optimize your extrusion process. And I'll talk about that at the end of the slide set and show how that can be done. And then finally, particle technologies. So these can exist in various sizes, micro or nano in scale, and then shape-wise, spherical or irregular particles are common. The system contains, for example, PLGA encapsulated around an API, so you can find these microspheres. The better image is this one from Zilretta. A PLGA matrix, the active is encapsulated within and throughout, so it's not just in the center or on the surface, it's throughout the matrix. So you form these particles, you typically store them dry and separate to the diluent. At the point of application, suspend those microparticles in the diluent, pass them through a needle, inject them into the patient. The polymer will swell, it will take up moisture, it will form pores, and the drug will continue to be released over the period of time that's been pre-designed. Again, pros and cons. Pros exist in terms of the needle sizes that are attached to these types of systems, so you can tune the particle size downwards towards small needle size. You can tune profiles again. Many products exist, so particle technologies would represent the largest number of products on the market in the space, but cons exist with respect to the technology in the sense that it's quite complex. It takes a lot of investment in terms of R&D to develop a process here and to be consistently able to produce particles with monodisperse size, good stability, prevent agglomeration, and get the release rates that you wish and target. So based on understanding those three different concepts, we can take a look at products on the market. So everyone listening should be able to understand that the three different products here, you know, what type of format they are, the first being an in situ forming system, Eligard, which delivers leuprolide acetate. It's a nice product to study actually for an explanation on how PLGA works, because there are 4 different formulation versions. So I'll show them in a follow-up slide. On the right-hand side, you have one syringe containing the PLGA polymer and NMP, and on the right-hand side, the leuprolide acetate. So at the point of application, these are linked, mixed over and back, loaded into one syringe, and then injected subcutaneously. So just as I showed in the video on the previous slide, the depot precipitates and the polymer is what will control the release over the period, whether it's a month or 6 months or in between. In terms of microsphere technology, Bydureon delivering exenatide for diabetes indication. So microspheres are produced and stored separately, lyophilized to the diluent. At the point of application, the microspheres are loaded into the diluent. So you see the window here with the diluent, and these are self-administered weekly by the patient. Exenatide happens to be the larger molecular weight compound that's been proven with PLGA type technology, so about 4,187 Daltons. There are technologies now looking at proteins, trying to work with these. We're working on projects in those areas, so if there are teams listening, working in those areas, we'd welcome discussions. PLGA type chemistry is quite broad, so we can do a lot in terms of customizing towards working with your compound, stabilizing it, and effectively delivering it. And then finally, solid implants. Ozurdex is a nice example. So an intravitreal dexamethasone implant. It's implanted directly into the eye. The PLGA then controls release of dexamethasone, treating the indication for uveitis. So based on those concepts, you could take a look at many products. So I show some here, just a sample from the market. I'm not going to spend time talking through each of these, but just to make a few things clear, the indications are typically chronic or long-term indications. So understanding PLGA type technology, you would recognize that that's exactly where the benefits could lie. A condition that's not going away today or tomorrow or this week, something that's very serious, where we need to be very strong in terms of how we treat patients and how we ensure patient compliance. These are represented by serious indications, things like cancer, diabetes, opioid dependency, parasite treatment. All of these are relevant types of technology, and the market is expanding. The number of applications is expanding. The number of technologies that are adopting and trying to work with these systems are expanding. And if you're thinking about your compound now, think about whether or not it could benefit from a weekly or monthly or bi-yearly or quarterly release system, and that will be a quick estimation as to whether you're relevant for PLGA type technology. The drug substances, some of them are shown here. The API sizes, many small molecules, several peptides, up to a maximum of exenatide and Bydureon. The dose that's relevant, so I show a sample here. If you were interested in a maximum dosing, a relevant question to ask, we could talk directly. So you can reach out to us and we can set up a phone call to talk about that. That's often a target. So what is the maximum that you can deliver? So that comes back to which format you're working with. Some have more potential perhaps than others, and we can give you advice on that based on what's been achieved in the market, as well as working with you to get beyond that if that is a target. And then finally, on the right-hand side, duration of release. So many systems doing a week-long release, 1 month, 3 months, or 6 months of release based on various PLGA technologies. So it should become clear as we talk through this that it's the PLGA technology or relevant bioresorbable polymer that you're using that actually allows you to achieve this 1 month, 3 month, 5 month, 1 week or so on target that you're looking for. And that's the part that we can really offer success and help with, tuning that, selecting the right chemistry and working to develop a commercial process. So we can move on now to controlling drug release via chemistry. And this is Ashland at the head in terms of what we can do for you. We can offer expertise on chemistry. That's exactly where we're focused. That's what we do day in, day out with these types of polymers. We work on many projects, a very good understanding of processing these types of chemistries, and the polymer alone is a system that we can very nicely control. So when we work together, we're taking into consideration your API, and there's a little bit of trial and error and working out, ironing out the issues in those types of formulations. But the chemistry is definitely something that you should seek to take advantage of with your supplier. So think about PLGA first. We need to understand the chemistry and where it comes from, how it works, and that will give us a good understanding of how it will behave in the body, but also how it should be handled in your labs and in your trials. We start on the right-hand side with the monomers that are used. So for example, in the case of PLGA, lactic acid and glycolic acid. If you respire anaerobically, so if you don't get enough oxygen into your muscles, you can produce lactic acid and cause a cramp. Your body knows how to deal with lactic acid. It can break it down through Krebs cycle into water and CO2. So we're taking advantage of something that's very safe and very inherently active in the body already. And as chemists, we build these monomers upwards from monomers into predictable chain structures. So this is a step where we would program a polymer for 1 week versus 1 month versus 6 months of release. We then supply that polymer to you as a formulator, combining it with an active pharmaceutical ingredient for your compound. You may place that back into the body in various different formats. And then via hydrolysis, so the addition of water breaking the ester bonds within the chain, these chain lengths of polymer will be broken back down over time into oligomers and finally back into monomers. And the monomers will be processed by the body via Krebs cycle, via excretion, and via other pathways back into water, CO2, and harmless products. So it's a very safe system. We're not adding anything that would cause problem in the body. We're using a chemistry that the body really well responds to and understands, and we're delivering polymers based on a programmable system that you can be consistently reliant on. The most important thing to take from this slide is hydrolysis. So everything that you do in terms of handling, so when you store it in a sealed pouch, in terms of processing when you work on an extruder, or in terms of sterilization or post-processing applications, you always need to be conscious and mindful of water and how water can damage these chemistries. So try and resist water, try and remove it from all process steps, and repel it where possible in your application from start through to finish, and you will achieve a better outcome and a more consistent, stable system. So moving on to how we actually designed the chemistry. So if you think about the applications you have, I give a straightforward example here of a particle technology. So this could be a microparticle, for example. You encapsulate the microparticle with the API, so it's existing on the surface and throughout the polymer matrix. You then take this microparticle suspended in a diluent and inject it into the body, for example. So the first thing you'll see in terms of release is a burst phase. Over the first 24 hours, release of drug that would be present on the surface into the diluent and subsequently into the patient. Thereafter, you would have phase 2 diffusion controlled release. So as the polymer takes on water, starts to swell, it will form pores. These pores will get larger over time and diffusion will occur. So drug will be diffused from the matrix outward and be released. And this is typically the longest phase of release. And finally, a third broad phase of release, which is very often not present. So if you think about this, the polymer over time in the body, let's say over weeks and months perhaps, is losing length in terms of chain length. It's losing molecular weight. So as it gets shorter and shorter based on hydrolysis, the molecular weight will reach a critical low. So at that critical low point, it will start to occur in form of mass loss. So it would crumble. And if that depot crumbles, any drug that would be remaining would finally be released. So the reason you don't often see this is because there's no drug present when that typically happens. So if all the drug is removed by the time it actually crumbles, there's nothing to be burst at that stage. And what we're trying to engineer is a solution that will get you your timeline in terms of whether it's 1 week, 1 month, or 6 months. That part is less complicated, but more importantly and more complicated, the phase of release and rate of release that will get you a concentration within the bloodstream or within your target area that's as targeted and as required. So that's what we really try to work with in the chemistry, the duration and the rate of release. So this comes back to chemistry. PLGA is a good example we can start with, and this one will be a nice explanation for this challenge. So lactide and glycolide. On the lactide side, D and L are the existing isomers of lactic acid, so DL lactide. On the right-hand side, glycolic acid, 2 repeating units to form glycolide. We use ring opening polymerization, so we open dimers to create that, which is not so relevant for you to know, but to be aware that that's where the technology comes from, and that's why it's called lactide as opposed to lactic in the case of a ring opening polymerization technology. Lactide is more hydrophobic or water repelling than glycolide. So if we create a polymer that's more hydrophobic, it should last longer in the body. And that's the first broad control that you have. You make the polymer more hydrophobic by increasing lactide content, and so it should last longer in the body. The second control would be the N group, or in this case, typical polymers exist with acid or ester terminated N groups. So a carboxylic acid N group, for example, is more hydrophilic or water loving than an ester terminated polymer. So if I want this polymer to degrade quicker, I would work with an acid termination versus an ester termination. And then finally, in terms of simple characteristics for degradation, the length of these chains, so the molecular weight or inherent viscosity, is important. The longer the chain length, the more hydrophobic the polymer structure is, and typically the longer degradation time would take. So based on those 3 controls, you could summarize very quickly and say that the longer acting polymer would have largest lactide, so 100% lactide, for example, an ester termination and a large molecular weight. So that's a very broad, simplified manner for saying what will degrade slowest, and we haven't talked about the API yet. And for the polymer that would degrade quickest, we should increase glycolide content, we should work with an acid end group, and we should work with a low molecular weight. So obviously it's a bit more complicated than that, but those are the basic controls that you have. When the API is introduced to the system and the formulation comes into it, some complicated features come up, things that we can work with. You just need to have the experience and guidance or support available to you. So when you work with these systems, we can help. We can offer support in terms of designing your system or some of the relevant questions that you have. So your questions may be what depots have been formed in the past. We've just talked about 3. What types of drugs have been used? What's the maximum drug loading that you have achieved in the past? Which system would be most compatible with our compound? We can help you in this area. Beyond that, we have the relevant components that you require for these types of systems. So Ashland have built a strong portfolio in this area. We offer volatile bioresorbable polymers. So these are the controlling polymers for the controlled release function. In the case of the in situ forming systems, we offer the GMP grade of NMP on the market under the trade name Pharmasolve. In the case of the microsphere technologies, Aquilon CMC is a very commonly used viscosity modifier for the diluents. We offer a bioburden and endotoxin tested grade. And then finally, Plasdone C grades are used in some situations for crystallization inhibition and in some of the in situ forming implant technologies in research to change the way the depot actually forms when it forms in the body to affect the release profile. So we offer all of the key components that you could require. This is obviously useful because you need those to formulate, but more importantly, we have very strong experience working with these because we have them in-house. We have many programs that we seek to use these together. So we have an extremely good understanding of our own components and how they work together. And that's an unknown that you would like to work past. So a supplier like Ashland can get you through that quickly, which might represent an acceleration in your time frame to concept. Beyond offering those components and support, we have advice that we can offer you, whether you're working with solvents or melt-based technologies. We've done a lot of work there. So for example, anybody on the call listening, if you're working with a solvent-based approach, are you aware of all the solvents that exist in terms of solvents that could be used with PLGA? Are you using the cleanest or nicest solvent for your system? Are you aware that polymer chemistry is linked to solvent compatibility? So some solvents won't dissolve certain PLGAs while they will dissolve others. Have you understood all of those aspects and can you benefit from some support in those areas? We can offer that type of advice. For the thermal processing aspect, I'll show you how we could offer support in those areas at the final slide. And then finally, post-processing, because these chemistries are very sensitive, sterilization and how you would complete that has its own unique set of rules and guidelines. There are 13 options that are typically talked about, of which 4 are commercially relevant. If you're interested in knowing more about those areas and the options that exist, or data on how vital polymers perform under sterilization, we can share those pieces of information with you if we're working together. In terms of functions to control release, so if we re-summarize some of what we've just talked about, broadly, the duration of release. So if you said you were targeting a week versus 6 months, we would have a very quick understanding of which chemistry you should consider and which monomers you should be working with in terms of the polymer structure. The ratio, we talked about lactide and glycolide and how they're important in terms of how they'll dictate what duration would be required. But beyond that, at a very micro level, let's say the macro structure is 50-50 lactide to glycolide. And so you have 50-50 molar ratio of both monomers in the structure. But at a micro level, in terms of sequence of distribution, have you understood properly what sequence your polymer is providing? So in terms of L for lactide and G for glycolide, is it LG, 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 and therefore being a Bernoullian random copolymer, or is it more blocky than that? So there are ways to characterize that. We offer support in that area. And what you really need to understand is that that will make a difference when you get to tuning out your process. And you need to know that your supply is consistent and you need to understand what your source is providing you in terms of those types of numbers and specifications. N group we talked about, and I have a slide that will get into a little bit more detail. So there are other reasons beyond how quickly the polymer will dissolve or degrade towards considering the type of N group. The inherent viscosity, so the larger ones will typically lead to polymers lasting longer in the body. And then finally, chemical structure. So we've only talked about linear PLGAs so far. There are other structures in terms of structure of the polymer. A glucose star polymer is used in a sandostatin product called sandostatin LAR, Novartis product. So that's used to control release of octreotide. And there are other structures that exist, star or dendritic polymers, as well as new polymers that we typically will and frequently will work on. So can we put the theory to the test? Can everybody here understand why different polymers were selected in the different versions of Eligard that exist on the market? So if we take Eligard, and by the way, everything that's shown is public information, it's all linked and it's all publicly available. So we're just showing the composition of the 1 month system on the top here. PLGA polymer with a 50-50 acid type chemistry, the relevant NMP solvent and leuprolide acetate. So going from 1 month to 6 months, if you take a quick look, you'll see that the amount of drug compound changes, so from 7.5 to 45 milligrams, and that's quite a logical change. So you need more if you're going to deliver over a longer time frame. But the important point that changes in terms of controlling release is the polymer. So at the 1 month system, you can see that it's a 50-50 polymer, which has a relatively large glycolide content. From 1 month to 6 months, we increase lactide, which is more hydrophobic, to 85. Therefore, this polymer should last a lot longer in the body. Second point, the end group is acid in the case of 1 month, which is changed to ester in the case of 6 months. Again, this should last longer in the body. The additional points that are not shown here are molecular weight and composition, so how NMP and PLGA are fractionally composed in the formulation, and you can expect it, and it is the case. PLGA composition will increase from, let's say, 1 month to 3 months. So there would be more PLGA in the formulation in the 3 month system. The idea is there that when you form this, it's going in solution and changing from solution to solid. So you need more of a net to capture the drug to reduce burst effect. So you load more polymer to make it more viscous and capable of that. And thereafter, more polymer would allow it to last longer and restrict release so that you don't have any issue in terms of burst and accelerator rates. So this is how you work with these types of chemistries, and everyone on the call should understand broadly at least why this polymer is relevant for a shorter acting system versus this polymer. The other thing to be aware is that polymers don't have defined release profile for every API. The API itself, the charge, the size, the type, and so on will make a difference. The formulation format will make a difference. So while we can say that we know that this polymer will release quicker than 8515, for the different APIs that exist, the time frame might be a week versus a month, as opposed to a month versus 6 months. And we design through that based on real data and developing real projects with customers. So some other information for everyone on the call in terms of other structures. So we offer GMP grade polymers, all of the standard types of chemistries that you've worked with so far, hopefully PLGAs, PDLLAs, polycaprolactones, polylactide type polymers, the various N groups, the various inherent viscosities and molar ratios. But beyond that, there are other chemistries that are being explored in the market, and we work in these areas also. So chain N group, for example, PEG addition, so PEG terminated polymer. PEG is extremely hydrophilic. It will gather a lot of moisture, it will gather it quickly, and it will cause a polymer like PLGA to degrade quicker if it was included in the formulation. A PEG N group could be used for that strategy towards tuning degradation of a polymer that would typically take a long time to disappear in the body. So for example, polycaprolactone could last several years, but if we create a PEG terminated polycaprolactone, it may be accelerated drastically so that it would degrade within 1 year, for example. That might be one strategy that you could consider. More complicated type strategy, for example, PEG terminated PDLLAs towards this micelle technology. So on the right hand side, you see PDLLA PEG as a chain structure. You have this hydrophobic hydrophilic interaction, so you have some amphilic charge. You can use this type of structure to create micelles where you line up the charge separation with the drug compound in the center to create these types of nanoparticles. And that's exactly what Genexol does, a technology on the market. Beyond that, different monomers could be composed. We could work with different polymers, new chemistries, various molar ratios that have not been used before. So for example, PLGA with 8119 or 8713, all of these things are within our power and we can supply them for you. Sometimes teams are interested in innovation for those types of technologies, but more often they're useful in design of experiment and setting out your specification limits. And then finally, the different structures. We've talked a little bit about the Sandostatin product, the Glucostar PLGA. There are other structures that you could work towards. In some cases, these might be useful towards large molecule release. So large molecules are a target for these types of systems. There are certainly challenges, but having this type of toolbox available to you in terms of customizing chemistry gives you a much better position towards actually solving and delivering a solution for extended release large molecule or protein type compounds. So for example, again, PEG is a very useful stabilizer, could be useful for stabilizing some compounds. It's a useful stealth molecule because of its flexible chain structure. It could be used to deliver some sort of stealth vehicle and so on. So process optimization, now that we've understood the products that exist on the market and how they look, the formulation formats, a brief on chemistry, we should now take a look at how you might process these types of polymers. So today's example is a hot melt extrusion example. But if you wish to work with these systems in atrogel type technologies with PharmaSolve solvent, for example, or with microsphere technologies, please do reach out. We have a lot of expertise and information that we can share. We can help you get from zero to your concept formulation quicker, certainly, and hopefully we can debug some of the issues that you might have moving forward. So a hot melt extrusion process, for example, if we're taking a look at this, for PLGA type chemistry, one of the obvious things that we need to be conscious of is the polymer degrades. So if we subject it to very high temperature and shear stress, we can cause excessive degradation. And if I degrade this polymer going through this process, it may not perform as I wish it to perform. So it might be designed towards 1 month of release, but it might only last for a week if I mishandle it. Second to that, the form of the polymer is really important. So not only for the basics, so for example, there are forms that exist for PLGA that are fluffy or low density that may have static charge. They present challenges in terms of how you might feed as an extruder. So volumetric flow and gravimetric flow, you would prefer to work with something that's denser and more powder-like so that it would flow into the extruder. And beyond that, again, polymer and drug dispersion. So it makes sense to work with powders that are similar in terms of size. If you think about the hopper and how it works, if I have a large particle of PLGA, for example, let's say rods, and if I have drug that's a powder form, when I load it into the feed, the powder will sink to the bottom and the rods will stay up high. So you'll get some displacement or problem in terms of how the drug is mixed. So your physical mixing step isn't giving you any advantage that you could get from it. So when we think about these things, how can we improve hot melt extrusion performance of PLGA? Dense flowing particles would be useful. How could we reduce degradation? So particle size would be important. If we increase surface area, we might be able to program the particles to require less energy to melt. So if they need less energy to melt, we should do less damage to the structures. And then dispersion of drug, again, if we can tune the particle size of PLGA towards being similar to that of the drug, we should be able to achieve better mixing before even entering the barrel. And that might be the difference between needing to extrude once versus twice. And if you need to extrude twice, you might need to do it for your mixing. And if you do so, do you degrade the polymer even more than required? So based on that, we developed different particle forms. So on the left-hand side, you see volatile polymers. These are PLGA's 50-50 acid-terminated polymers in a powder form versus a granule form. Roughly the size indication about 350 microns average particle diameter versus about 3 millimeters average particle diameter, so a 10x increase in particle size. We did this work with Leistritz based in Germany on their 12 millimeter extruder. So Leistritz are an excellent team to work with. Technology is outstanding, a lot of flexibility, very able to customize and tweak as needed. And we worked over these studies to try and optimize how PLGA could be processed, because we recognize that many are finding issue with this and perhaps could do with benefit in terms of the particle form towards a more consistent process. We ran several runs where we optimized by tweaking screw speed, feed rate, polymer type. So we just say fine versus coarse granule. It's the same PLGA. It's just different format, and temperature profile. So temperature profile is much more important rather than just saying temperature. Temperature at the die head is typically at the maximum, so 130, 110 and 90 degrees Celsius, but we're ramping this up from zone 1 up through zone 8. So we studied all of these things to try and understand which polymers work best in terms of processability, flow rate and so on, which allow for lowest degradation and which will give us the best output. And to summarize, the powder form that we supply is absolutely outstanding for this type of application. The coarse granule works okay, but it can lead to some frustration, some headaches in terms of blockage, clogging, material flow, air pockets and so on. But the powder will work nicely with gravimetric or volumetric flow. There's no blockage. We didn't observe any loss based on blockage or clogging, so you've less downtime on the equipment. In terms of mixing compounds, the particle size of powder allows you better flexibility in terms of mixing with your drug compound. And then more importantly, studying the chemical characteristics through the extruder. Top right-hand side, coarse granules, so the larger particle size, you can see that we observed at 130 degrees Celsius profile about 15 to 18% reduction in molecular weight. So it's common to lose some molar mass in extrusion. It's very common. You may have to accept that in your process, but this certainly should not be acceptable to a process like this. So we optimized it for the coarse granule, and then we found that when we worked on the fine particle, we could optimize this much more significantly, so down to about 5% reduction in molecular weight. And the reason for this is if you work with a powder, it's got a larger surface area, so it melts quicker. You do less damage because you apply less energy to melt it. When it's melted, it's in a molten state, so there's less shear in the barrel. So altogether, these things combine to lower damage to the polymer. And the other point behind that is at the higher temperature, there's lower viscosity, so you may be able to target a better dispersion of drug at these higher temperatures with this type of polymer. So the summary on this is that particle form is very important for hot melt extrusion, and behind that you need to do the studies to actually understand what types of changes are being observed. So this polymer would not perform similarly to this polymer, even though you've used a hot melt extrusion process on both. The only difference being the input was a coarse granule versus a fine powder. So second to last slide, I think some common challenges. Chemistry-wise, we can support you on selecting polymers, but also on how you may optimize them. And then when you're working with these systems, I mentioned earlier solvent-based technologies. Right inside you see an image showing solubility of some polymers in various solvents, but the lactide and glycolide composition actually makes a significant difference. High glycolide is very insoluble in most solvents. High lactide presents very little issue for most solvents. So we can provide data on which specific ratios will be soluble in various solvents so that you could effectively clean out your process or work with solvents that you may not have been aware of. So some solvents with better regulatory compliance or certain solvents that may lead to more homogeneous particle distribution and so on. Melt-based technology, we can share more data than what I showed today if we're working in projects. So I showed today what we publicly share. If we're working together, we can give you much more support in terms of start to finish through how you would optimize, including some application studies on optimizing your system. And then beyond that, post-processing, things like sterilization, we can offer the existing options that exist, the pros and cons, typical approaches, and some guidance on what would happen to volatile polymers based on the sterilization options that you work with. So we have data in those areas. So the second last slide, I think, just on how we work on this, we optimize samples. So for example, if you know what you need, that's great. If you don't, we can suggest. We supply some samples. You may do some analysis of your own process trials, applications testing, and figure out, for example, you got 1 month of release when you were targeting 2 months of release, something like very simple like that. And in that way, we can optimize around the polymer that works best for you. And over time, we can optimize quickly with you to refine the specification towards the optimum target. So many grades available from Ashland. There are 48 products shown on screen. All of these are GMP grades that we keep on stock typically. We have a type 4 DMF filed with the FDA. The site is ISO 13485 compliant for the quality management system, and everything is manufactured within a Class 8 cleanroom. Beyond these polymers, we also do made-to-order specifications, so any tight windows that you have, you can work with us on those. We're very flexible. We're flexible on form, and we do custom polymers outside of these chemistries. So with that, I would say thank you to everyone, and if there are any questions, we can talk through those now.
Thank you very much, Sean. We will now move on to the Q&A portion of today's webinar. You can still submit your questions during this session, so you can do so by clicking the Q&A icon at the top right of your screen and submitting your question. So we'll start with our first question. How do I adjust release for an in situ forming implant based on NMP and PLGA? Can you share more information on these types of formulations?
Yeah, and that's a really excellent question. So the presentation today was focused on hot melt extrusion. We probably have even more experience with the Atradel type system because we offer the NMP and the PLGA, a lot of studies and data. So we could set up a call if you want to send me an email to go through it in detail. So a lot of slides and detail on how that can be done. It starts with chemistry and some of the rules that we talked about apply, but very importantly, the composition of PLGA versus NMP and how you formulate that in your depot is extremely important. Some other things come into play that are not common in the other depot forms relating to molecular weight. So because you're changing from solution to solid, you have to be conscious that that phase inversion stage can make a big difference in terms of how the depot forms. So going from liquid to solid and how porous that will be will dictate a lot in terms of how the compounds are released thereafter. So if you are interested in that type of data, please do send me an email and let's set up a meeting to talk through it in more detail.
Great, thank you, Sean. Our next question is, what is the lead time for a made-to-order or custom polymer request? How can you get a sample?
Yeah, so if it's in stock, it's straightforward. So it's the lead time that our supplier allows. So it's typically 3 to 5 working days, depending on how far away you are from the manufacturer site, which is based in Ireland. If it's a made-to-order polymer, it depends on scale. So we have many different reactors for different scale sizes in terms of batches, but typically 2 to 3 weeks for a tweak to ratio, N group, or molecular weight. So if you wanted a PLGA with a unique ratio, N group, or molecular weight, 2 to 3 weeks is quite typical. So we're very fast in that. And that comes back to our technology, many different reactors available to offer customized batch sizes, all of which comes from within our GMP facility. And if you wish to get samples, you can reach out to your sales rep within Ashland, or you can send me an email directly at the email on screen.
Okay, our next question is specific to slide 8. So okay. The question is, what solvents are you referring to here? Looking at PLGA solubility, are these solvents safe?
Yeah, so that's an excellent question. And again, if we're working together, we can share all of these types of data. So there are about 25 solvents that you could consider. I'm not going to list 25, but for example, people might be familiar with solvents like dichloromethane, ethyl acetate, benzyl alcohol. THF tetrahydrofuran, all these types of solvents, but there are many more, and there are many that are considered higher and lower classification in terms of regulatory acceptance. So we can show and share data in terms of tables showing composition by ratio and how well it solubilizes in the various solvents, if that's interesting to you. So please, again, reach out and we can share that data and perhaps help you on your project.
Okay, our next question is, how do molecular weight and inherent viscosity measurements correlate for PLGAs? Can you work with both?
Yes. So yes is the answer. Again, very good question. We can work with both. The really important thing I would say about that is be aware of the test method, the concentration that's used, for example, in the IV method, the temperature and solvent that are used. But with respect to molecular weight, be aware of the calibration standards, the solvents that are used, the temperature, the type of equipment. So it's typical to use GPC. So we can see the same polymer giving 10 different results on 10 different pieces of equipment. So that's not so nice to try and work past. But if you're conscious of the test parameters, you can work past it. And with respect to correlation, you can certainly plot IV versus molecular weight. You can do it for IV versus MW by weight average, or IV versus MN by number average molecular weight. But it's important that you do that per ratio. So a 75-25 PLGA will have a certain IV versus molecular weight slope, while a 50-50 polymer might be slightly different. So if you want to do that, we can share some data, or if you have polymers of your own and you're really interested in doing that, we might be able to support you with that type of study.
Okay, our next question is, can we select narrowed inherent viscosity grades? What tolerances are you capable of supplying?
Yeah, and again, a really good question. So it is extremely important to be concise and to be accurate in terms of how the polymer will perform. If you're delivering something over a week, can you afford to have a tolerance in terms of hours or days? And a lot of this comes back to the spec on the polymer. So yes, we can narrow the spec. We do so from two options effectively. So we can do lot selection, and based on lot selection, you may be able to find exactly what you want every time. So if it's 0.20 as the target, if that's in stock, we can supply that to you directly. But if it's a made to order scenario, we can work extremely tightly. So our typical brochures give a broad range because we have to indicate something. But in terms of narrowness, we could probably work to 0.2 plus or minus 0.02. So 0.18 to 0.22. And if it was absolutely necessary for you, we could tighten beyond that, so we could make it more consistent as needed.
Our next question is, how are PLGA-based formulations sterilized for pharmaceutical use? Are there any side effects that we should be concerned with?
Yes, and again, really good question. So there certainly are side effects that you should be concerned with depending on the method that you used. So I mentioned earlier that there are about 13 options that you come across when you read about this. If you like, we could go through the 13 options and talk about the ones that are relevant, but of those 4 relevant options based on irradiation techniques, so gamma irradiation or electron beam irradiation, they're quite commonly used. Ethylene oxide may be used, and it's often used, or sterile filtration. So those are the 4 options that I would suggest considering if it's possible to use them. Sterile filtration is a very nice option in that when you're sterilizing via that option, you don't apply any stress to the polymer, so you don't cause degradation. However, not all systems can pass through the micron filter, the 0.2 that's required, so very often that might not be an option to you. And many teams then revert back to gamma irradiation. So if you do work with gamma irradiation, it will degrade the polymer typically. There's a certain amount of degradation, and that depends on the molecular weight and structure of the polymer. So we can work with you on assessing that for your actual formulation, and even before you get to that stage, we can show you data on volatile polymers and how they're impacted when you sterilize. And the obvious advantage there is if you're designing a system and you know that your polymer works perfectly, and you know that you're going to work through gamma irradiation at the end, it would be wise to build in a tolerance in your process. So plus 10%, for example, if you know that 10% is the reduction that you'll expect from irradiation.
Our next question is, do you have any applications on preclinical animal study?
Yes, so depending on the case, yes, we do. We've done several projects in terms of animal studies that we've worked with customers, as well as projects that we've done ourselves using these types of polymers for various different drug delivery and medical device type applications. So again, if you want to know more information about those areas, we can share if we're in direct contact.
So our next question is specific to slide 6. And the question is, what is the mechanism of drug release through PLGA matrix? How does the concentration drop for other conventional PLGA polymers, and how does Viatel take care of that?
Yeah, so I think I understand the first part of the question anyway. So what is the mechanism for release? So to answer that, The first thing that you should be aware of is that the polymer has a degradation rate, and that is very much set to the polymer. The polymer is programmed to degrade in a certain way. So you can program the polymer, you can do an in vitro or in vivo assessment, and the polymer will degrade as it degrades based on how we form the chemistry. When you put a drug in combination, be aware that different drugs will behave differently, and the release of the drug is linked to degradation of the polymer, but it's not exclusively linked. So there are certain cases where the drug will be released based on the degradation rate, but it might be different for different compounds. So degradation rate is the first point to be aware of. Typically, degrading polymers that degrade quicker will release drug quicker. There are other things going on behind the scenes in that type of case. So for example, when you put the depot into the body, it takes on moisture and it swells. If you have a compound that's more hydrophilic or hydrophobic, that will obviously be impacted. The size will be impacted. So if you're forming pores in the polymer matrix, if the pores that are at a certain size in your compound is small enough to pass through them, you will have release of the molecule through those pores. So there are several stages of this. So you place the depot into the body, polymer swells, takes on moisture, starts to degrade, pores form, diffusion of the active starts to pass through, molecular weights decrease over time, polymer continues to swell, pores get larger, polymer allows diffusion of molecules, and over time all of the depot would be released. Thereafter, it's common that all of the drug would be released by the stage that you reach a critical molecular weight. The depot starts to crumble and physically breaks down. So there are very detailed explanations in the literature of many steps in terms of everything I've just talked about, but those are the common ones that we think about when we're working on a formulation.
Okay, so our next question is, when using NMP, is there a concern regarding the toxicity of the solvent? Could you provide information about the maximum tolerable amount?
Yeah, so again, that's a very good question. So I would say there are 5 products on the market as recently as I think last year, a new one entering the market that use this type of system, many for human use, 1 of them for animal and 4 for human. So from those products, you get a very quick understanding of what's been tolerated in the body. The maximum, I think, is the Sublocade system, the 300 milligram dose, which delivers 833 milligrams of NMP in a single injection. The full injection volume is 1.5 ml. So that's a nice example. In terms of regulatory compliance, you can reach out to Ashland and we have a statement on acceptability for NMP that we can share with you.
Our next question is, what is the relative influence, if any, of D versus L lactide as it pertains to release profile? Does choice of stereoisomer influence the end result?
Yeah, again, that's a very specific question and a very good one. In the case of PLGA type technologies, it's very common to use a racemic mixture, so D and L at 50-50. So you don't often have, not that you don't have control, you don't often seem to come across polymers where you would try to influence that in the drug delivery space. However, in the medical device space, it's very common to work with an isomer, so PLLA, poly L-lactide exclusively, as little D lactide as possible towards crystallinity. So the reason we use D and L are they're isomers, so they're like your left and your right hand, and when they fold together, they don't properly fold and allow you to form crystalline regions. And that's important for drug delivery, and that's why we use a racemic mixture for drug delivery. If they don't fold properly, they don't form crystalline regions, and that's what means the polymer would be amorphous. Therefore, it breaks down predictably. So you would not commonly apply that type of technology in pharmaceutical drug release. It is, however, relevant for medical device industry where you're looking for very large crystallinity. So you want an isomer, L-lactide, for example, and 100% if possible, towards a large crystallinity and a better performance in terms of some physical attribute. So it's not so relevant for drug release. If it's a target for you, we can talk about it, but it's more relevant for medical devices.
Okay, our next question is, HME process is not feasible and available in our country's pharmaceutical companies. Are there other processes available that will have the same effectiveness?
Yeah, so in terms of effectiveness, you have to be aware that there are different technologies, yes, certainly, but will they perform as a hot melt extruder would? That's debatable because of the shear, the profiles and so on that go on in extruders, but more practically, extruders are very useful for commercialization. You can run large amounts of material through extruders and actually commercialize a technology based on extruder. If you simply wish to use a melt-based technology, 3D printers are a very good option to work with. You can melt polymers, pass them through 3D printers, you can incorporate drugs within them. The systems are widely available. There are systems that are available that print volatile polymers. They can take powder inlet. They can also take filament inlet. So certainly, yes, you could work with 3D printers. However, you have to think about what you're trying to scale towards. Will a 3D printer be acceptable for you in terms of mass manufacturing your commercial product? So that might be a debatable point. With respect to the extruders themselves, there are many large extruders that are quite expensive, but there are also smaller scale extruders that may be viable for these types of technologies. So you could work with, for example, micro extruders. So there might be some in-between space that you could consider working towards.
Okay, our next question is, how is the release profile measured analytically during development for these systems? And can you provide support or help with this?
Yeah, again, very good question. So we can provide support. Our director of formulation has a lot of experience in these types of systems. So there are 7 USP type apparatus that you could work with. Of those, USP type 1, 2, 4, and 7 are recommended for these types of systems. The literature indicates USP type 4 as being the most optimal for the system. So depending on the depot that you're working with, if you have option from 1, 2, 4, or 7, that's what you would try to work towards. There are standard methods for working with these systems, and yes, we can provide some guidance in terms of how to set up your system and how to set up your dissolution apparatus.
Okay, and we have about 5 or 6 more questions for today that we'll try to get through here. So the next question is, you provide the viscosity for different molecular weight. Can you also provide molecular weight ranges?
Yes, yeah, so it's a good question again. When we provide a COA, we typically provide inherent viscosity, and that's what's required. That's required from the regulatory guidance for us. We always test every single batch, no matter what, via GPC, so size exclusion chromatography. From that, we have the weight average, number average, and polydispersity index measurements for every batch. We can provide you that information, so all you would need to do is ask for it, and we can provide it to you. So per batch, we can provide it. If it's the case that you're working exclusively off molecular weight, we can work to that with you. So again, I would just reiterate that it's important that we can share the method that we have and the method that you're working with, so that we're working off the same method effectively, so that when you ask for 40 to 50 kilodaltons, that we give you a polymer with 40 to 50 kilodaltons from the same method, if that's what your target is. So yes is the answer, and you would only need to ask.
Is there a lower limit temperature at which hot melt extrusion can be performed for PLGAs?
Yeah. That's a very good question. So certain compounds might be sensitive to temperature, and yes, there will be a lower limit. So melt temperature of the polymer might be the limit. If it's not possible of melting it, you would need to be above that, obviously. So the study we showed earlier, 90, 110, and 130 degrees Celsius were what we used. You could reduce below 90, so I'm not sure what the maximum low would be in that case. You could probably work at 80 degrees Celsius, but you certainly need to melt the polymer, and the polymer PLGA, depending on molecular weight and ratio, will typically melt in the range of 70 degrees to 100, so in around 80 to 90 degrees Celsius. With that said, if it's a target for you to try and develop a polymer that will melt at a slightly lower temperature and perform as PLGA does, we have options. So there are several chemistries that we can work with towards reducing the melt temperature of the polymer, while also allowing you to release over time. So for example, we have a project where we're looking at polymers that will melt at around 60 degrees Celsius and degrade there afterwards, allowing the same type of function for a compound that is thermally sensitive in the number range that we just talked about.
Okay. Our next question is, release from PLGA is typically sigmoidal. Do you have any strategies for getting zero order release kinetics?
Yeah, that's a good question. And again, for that type of question, it really will depend on your molecule and your format. So an implant versus an in situ forming system versus a microparticle will each have their own constraints. Your molecule itself will make a significant difference what the size and charge and hydrophilic, hydrophobic behaviors are. So we do have strategies towards achieving that. There are strategies towards program the polymer so that it performs in certain ways when you load it into the body. These come back to all of the mechanisms that we talked about in terms of chemistry, as well as the format itself, so surface area, how you actually load it into the system, pre-treatments that you can apply to the polymer to try and remove drug from surface if burst release is the issue that you're concerned with. So we can work with you on that, and yes, there are mechanisms that you can use to tweak the release profile towards the target.
What is a typical loss on viscosity after sterilization after gamma radiation?
Yeah, so on that one, the higher the molecular weight that you're working with, typically the larger the loss. So if you're working with a high molecular weight number, you will typically experience larger loss. So it depends on the polymer that you're working with. But for example, for a low to medium molecular weight PLGA, it may be typical to lose about 10% number average molecular weight. So rather than taking that number as anything useful, you should contact us and we can try and work with you with the data that we have for various polymers to show you what gamma radiation at particular dosing, so it really does depend a lot on how you've dosed it, will behave. So depending on how you've dosed your system and depending on the molecular weight, you'll get a different output. But 10% is quite a typical number that you'll come across.
Okay, our next question is, what is the maximum storage period for PLGA particles, and at what temperature is better to store them?
Yeah, so it's quite common for microspheres that are developed to be stored in fridges, so 2 to 8 degrees Celsius. That's probably the most common that you'll see from products on the market. Stability of these types of polymers, so from our experience, would work best in colder conditions, and that's why we package them in sealed containers, triple layer packaging, and store them in a freezer typically. So when we offer storage advice, we typically say for maximum shelf life, store at less than or equal to minus 15 degrees Celsius. There is some impact depending on the polymer that you work with, so that would be our advice. For particle technologies, it's much more common to work and store them in a fridge, and the duration of stability will depend on the polymer that's being used and the API molecule that you're working with. So if you want more information on that, you can reach out to us for some stability data.
Okay, and this next question will be our final question for today. And it is, does the rate of PLGA degradation depend on the location of the subcutaneous injection location on the body?
Yeah, so that's again a really, really good, really specific question. So it does certainly, yes. So thinking about PLGA, keeping in mind that it will degrade via hydrolysis, the inflow of water. If you inject into subcutaneous tissue that's more water loaded or versus that fatty loaded, you'll have some impact. So there are studies showing that fatty tissue will result in slower release versus vascularized tissue. And with respect to subcutaneous tissue, for example, for longest release in the animal type market, they will inject subcutaneously behind the ear. So it's a version, or you could inject somewhere else in the body in a fatty tissue. So it really will make a difference depending on the type of tissue that you're injecting in, subcutaneous as a group, but more specifically, what type of fatty level and what type of vascularization exists in the area that you're developing your formulation for. So yes is the answer, and it will make a difference. It will come down to whether the tissue has got vascularization and good moisture flow versus fatty, which would be slower.
Okay, great. Thank you, Sean. And thank you for today's very informative session. For our attendees, if you have any other questions that we did not get to today, or if you think of questions after the webinar, you can contact Sean, or you can reach out via the Ashland website. And you can also do so to request a copy of the presentation. So we'd like to thank you all for attending today, and we look forward to sharing our solutions with you at our upcoming webinars.